Hello, my name is Liz Quinlan and I'm a PhD researcher here at the University of York. My research focuses on Atlantic salmon and their exploitation throughout the medieval period across the North Sea Basin area. I'm a zooarchaeologist, meaning that I study the animal remains left over at archaeological sites in order to better understand how humans in the past interacted with their environment. I work here at the University of York's BioArc lab, where we have hundreds of reference skeletons that I and other zooarchaeologists use to identify animal bones from these archaeological sites. Now, my research project is part of a wider training network called the Sea Changes ITN, which is funded by a Marie Skłodowska-Curie action through the European Commission's Horizon 2020 program. This means that my project on Atlantic salmon is connected to 14 other projects on human-environment interaction, specifically focusing on the exploitation of ocean and freshwater resources across Europe and through time. We're studying everything from whales to walruses, as well as sea turtles, grouper, herring, cod, and of course, Atlantic salmon. Now, a major part of my salmon research involves the fish bones that were recovered from excavations at numbers 16 to 22 Coppergate, here in York. This was done between 1976 and 1981. These fish bones are housed temporarily here at BioArc Labs, and a colleague and fellow Sea Changes researcher of mine, Katrine Dirks, is also working with these Coppergate remains. She's looking at the flatfish that were in the collection. We both arranged to borrow approximately 30,000 fish bones from the York Archaeological Trust, many of which are contained in the boxes you can see right behind me here.
These 27 boxes contain everything from cranial bones like dentaries and clythra from huge cod to the tiniest of herring vertebrae and prootic bones. Using prepared modern bones as a reference, like the ones you can see in this clip, zooarchaeologists can study these leftover fish remains to understand more about the diet, economy, and social status of a site. These two groups, herring and cod, are actually the most common fish bones found at the Coppergate site, which is no surprise. Cod and herring were both incredibly important fish to the inhabitants of the site, which included Roman, Saxon, Norman, English, and of course, Anglo-Scandinavian communities. When the Coppergate site was home to the Anglo-Scandinavians, it was known as Jorvik. As a settlement between two rivers, the Foss and the Ouse, and with access to the North Sea coast, Jorvik was a great place to eat fish. The rivers had freshwater species, including those from the cyprinid family, like bream, roach, and crucian carp, as well as freshwater eels, and another important salmonid species, the brown trout. The nearby North Sea provided gadids like haddock, ling, saithe, and cod, as well as herring and several types of flatfish. Moving between those two places were the Atlantic salmon. Salmon are anadromous, meaning they spend part of their life in both saltwater and freshwater. Atlantic salmon eggs are laid in shallow, gravelly areas of a river called a red, which is circled in this image. After hatching, the juvenile fish spend much of the first few years of their lives in deep, cool, fast-flowing rivers, like the Ouse, and then migrate out to the North Sea. When they make this migration, they undergo what's called a smolting process, where they go from being exclusively freshwater fish to being able to live in salt water. The juvenile smolts will travel through areas like the North Sea before reaching the deeper ocean, where they spend their adult lives until they are ready to reproduce. The salmon return back to the same river they were born in to spawn, and the next generation continues the cycle. Atlantic salmon were historically found all over northern and western Europe, originating in rivers like the Thames, the Rhine, and the Elbe, as well as their many hundreds of smaller tributaries. Humans in Europe have been fishing for Atlantic salmon for at least 50,000 years, and the inhabitants of Jorvik were no different. Here in York, the Ouse is an historic salmon river, and while the current salmon population in the Ouse is quite small, we believe that they were far more plentiful during the Jorvik period. One of my research goals is to understand exactly how important salmon were to the Anglo-Scandinavian people living in Jorvik, and to try and spot periods of decline in salmon exploitation. But why study salmon at all? Well, these fish are an interesting way to connect modern-day fisheries, aquaculture, and conservation efforts with historical fishing and the past environment. Today, Atlantic salmon make up the majority of farmed fish in the United Kingdom, with well over 1 billion pounds in profits each year. In the medieval period, salmon were so prized that in some areas, only those who owned access to a salmon river could legally fish for them. And salmon were subject to high taxes. They could even be used to pay your tax. If you owed money for goods, you could pay it in salmon. Salmon were extremely important in Christian medieval contexts, with monasteries and abbeys often being given the exclusive right to fish for salmon in some areas of Europe. This makes sense when you think about it, as there were upwards of 150 days of the year that devout Christians were supposed to abstain from meat in the medieval period, and salmon were some of the most desirable fish, mostly for their high fat content and delicate flavor. Pictish petroglyphs, or carved stones, were made depicting salmon all over northern and eastern Scotland during the 7th to 10th centuries, giving us an idea of their importance in Pictish culture. By the 12th century, in some areas of Scotland, there were even laws introduced to limit fishing of salmon that may be seen as early attempts at conservation, perhaps due to declining salmon stocks in the local river. But these are all Celtic, English, Saxon, or Norman contexts. What about salmon in the Viking, or more accurately, Anglo-Scandinavian, areas of the UK, like Jorvik? Literary evidence for the importance of Atlantic salmon for the Anglo-Scandinavians is fairly scarce, at least in terms of what has been published in English. We do know that, like in the later medieval periods, salmon were considered a high-status fish by Anglo-Scandinavians. Fish weren't necessarily religiously important to the Anglo-Scandinavians until the widespread adoption of Christianity, but salmon appear within sagas and Norse myth throughout time. The god Loki was said to be able to transform himself into a salmon when he needed to escape after playing one of his tricks. Salmon are also one of the fairly common archaeological bones found at Scandinavian sites during the height of the Viking Age, from the 8th to 11th centuries. It's likely that the Anglo-Scandinavians in York used various types of tools to catch salmon, with archaeological evidence for net weights, baskets, fish weirs, and hooks found at Coppergate. This is represented here in this Jorvik reconstruction of two fishermen mending nets. Research by James Barrett, Alison Locker, and Callum Roberts suggests that there may have been even more salmon in the Viking period than in later periods. Some theories about why there may have been a decline in salmon numbers from the Anglo-Scandinavian period to the later Saxon and Norman periods include environmental changes due to extensive damming of waterways, overfishing due to the increase in Christian feast days and elite demand for salmon, as well as changes in fishing technology. As I mentioned earlier, one of my research goals is focused on looking at the environmental, social, and economic changes across the whole North Sea basin area that may have led to dramatic changes in salmon populations. The best way to assess this, along with searching through medieval records and literature, is by looking at archaeological salmon bones. A major problem encountered by zooarchaeologists is how fragile fish bones can be, and Atlantic salmon are some of the most fragmentary and delicate bones found at archaeological sites. Now, we don't know for sure why salmon bones don't survive as well in the ground as other fish like cod and herring. It may be related to the high fat content of the fish, acidity levels in the local soil, or even osteological reabsorption, which means that the fish may find itself needing essential nutrients during its long journey back upriver to spawn. To solve that problem, the salmon might absorb minerals from its own bones to supplement these nutrients, leading to thinner and more fragile bones overall. Whatever the reason, it means that salmon bones don't tend to survive well within the archaeological record. And finding sites with a large amount of salmon is especially important for studying past use of the fish. Coppergate is an unusual site because it has a relatively huge amount of surviving salmonid bones, at least 325 bones at the last count. The most common bones are vertebrae, as they are some of the densest bones in the salmon skeleton. Nearly 98% of the salmonid bones at Coppergate are vertebrae, and I've taken a further subsample of about 60 to study further. When you compare that sample of nearly 60 bones to sites that I've reviewed from all over the North Sea Basin area, many of which have less than 10 identified salmon bones, it's clear that Coppergate is an incredibly rich resource for studying these fish. But now that I've managed to identify potential salmon bones from Coppergate, you might be wondering what my next steps are. Well, the first thing any zooarchaeologist must do is identify what kind of bones they have. In many cases, we can do this just by looking at them, using cleaned skeletons as a reference to compare individual bones and find a match. We also use a lot of books and photographic resources and study for several years to learn more about the unique structures of bones. Fish bones can be particularly difficult, as many of the commonly found bones don't vary much between different species. This means we need to try to spot small differences, and sometimes our eyes just aren't up to the task. Atlantic salmon and brown trout are both from the same genus, Salmo, which means salmon in Latin. The genus, just like the family or the species, is one of the organizational units that scientists use to classify animals. When you're looking at bones, it's sometimes the case that the physical differences between members of the same genus are so small that at first they don't show up at all. This is the issue with some bones found in salmon and trout, especially in vertebrae, which, if you remember from earlier in this video, are the bones we find most often on archaeological sites. Here I've got two photos, both from the University of Nottingham's fishbone resource, with the vertebrae of an Atlantic salmon on the top and a brown trout on the bottom. Looking at them, they both have this very lovely honeycomb patterning in the center, with delicate bone that isn't well ossified, meaning it's not as dense and thick as the bone found in the vertebra of fish like cod or haddock. When looking at archaeological examples of these bones, we can sometimes make an educated guess about which species they are based on size, as trout are generally smaller than salmon. But as I've done my PhD work, it's become very clear that we can't always reliably tell the difference, which is one of the reasons why I'll be using biomolecular analysis to identify these bones and learn more about the environment they came from. Luckily for my project, we have a fairly simple chemical test called zooarchaeology by mass spectrometry, or ZOOMS for short. Now, bear with me for a second. I want to explain a little bit about the structure of bones. All bones are mostly made up of a protein called collagen, combined with a mineral called hydroxyapatite. The interesting thing about collagen is that its structure is a triple helix, and a team of biologists and archaeologists figured out that if you unwind that 3-strand chain and break it into smaller packages called peptides, then they can be used to identify sequences that are unique to a single species. These peptide sequences, or markers, are identified using instruments like these, called mass spectrometers, which help us identify differences in the mass of the peptides. The unique identifiers for a single species are known as a molecular barcode. And in 2011, a team of biologists, archaeologists, and chemists published the first barcodes for fish species, including those for Atlantic salmon and brown trout. So to do this ZOOMS analysis, I'll be breaking off tiny pieces of bone from the salmonids I've collected from Coppergate, and then putting them into hydrochloric acid to break down the collagen. Then I'll extract that collagen before breaking it up even further into the peptide sequences using an enzyme called trypsin. This analysis will hopefully give me a clear answer about the species of the salmonid bones I've collected from Coppergate, letting me estimate more clearly the proportion of salmon and trout at the site. The collagen I extract won't just be used for Zoom's analysis. I'll also use it for another set of biomolecular tests, which involves stable isotopes. Bulk stable isotope analysis of bone collagen basically means that I'll be looking at the ratios of certain chemical element variants, called isotopes, that are found within bones, and comparing those ratios to known isotope ratio features of past environments. You might be familiar with the carbon isotope carbon-14, which is an unstable carbon isotope, which can be used to determine the age of archaeological material. Well, my isotope analysis will involve stable isotopes, which don't decay over time, like carbon-12 or carbon-13, nitrogen-14, nitrogen-15, etc. I'll be doing two kinds of stable isotope analysis. One is more general and will hopefully tell us about the environment the salmon lived in. Things like how salty the water was they lived in, ocean temperatures, what they ate. This is all possible because these stable isotopes are recorded in the fish bones as they form during a salmon's life. This gives us what you might call a molecular snapshot of environmental conditions and helps us understand more about the fish that the Anglo-Scandinavians at Jorvik were eating. The other type of isotope analysis I'll be trying to do is called incremental stable isotope analysis, which basically means I'm going to try and extract collagen from the very center and the very outside of large salmon vertebrae to try and see if there is any difference in the isotope profiles and, by extension, the environmental conditions. Fish vertebrae form like trees, with rings of bone laid down over the life of the fish, and fish never stop growing. So theoretically, the bone from the center of the vertebra should tell me more about the freshwater or river environment that a salmon grew up in, and the bone from the outside edge should tell me more about the ocean. Now, this is something I'm really interested in understanding because it might tell us more about where the residents of Jorvik were getting their salmon from. Were they fishing in local rivers like the Ouse and the Foss? Or did they have to import salmon from faraway sites like Kaupang in Norway or Ribe in Denmark? If they had to import salmon, that might indicate that the local salmon population had declined. Since I'll know some information about the local environmental conditions from the time period, I can try to get a more clear picture of why exactly those salmon stocks may have declined, if they did at all. Now, this brings me to the wider relevance of my research and its possible connections to modern day ecology and conservation efforts. Because the real question here is, how can fish bones from 1100 years ago answer questions about salmon today? Stocks of wild salmon in the UK have been steadily declining since the 1970s, even as conservation efforts in individual salmon rivers have increased. There are a variety of reasons offered for this decline, including global warming, overfishing, disease, but no clear single reason. Organizations like the Atlantic Salmon Trust, the ICES Working Group on the Atlantic Salmon, and the North Atlantic Salmon Conservation Organization, or NASCO, are all concerned with the steadily declining numbers of salmon found across the North Atlantic. This map on the left of the slide, taken from NASCO's 2020 report on the state of the North Atlantic salmon, shows that there are very few salmon rivers in the United Kingdom which have sustainable levels of salmon, despite years of restrictions on salmon fishing and studies on individual populations. Hopefully, the information that I get from conducting my isotope research and reviews of Atlantic salmon use during the medieval period will give me some insight into the environmental and social conditions that led to the collapses of salmon populations in the past. I will hopefully be able to tell conservation scientists what environments were like in, for example, the year 870, when Vikings lived in York, and how that information and the number of salmon found at the site changes over time, and if those things might be related. Then, when conservation biologists are researching why salmon today are struggling, they have some historic contexts for the rivers that they're working in. But even if these modern day applications are minor, the information I'm hoping to learn will still be incredibly valuable in terms of our knowledge of the Anglo-Scandinavian period here in York and the Scandinavian period in general. The results that I get from my research on salmon during the Viking Age in Jorvik will be compared to other famous Viking sites, like Dorstad in the Netherlands or Kaupang in Norway. I hope that by learning about how people in different settlements exploited salmon during the medieval period, that I'll be able to understand the broader trends of trade and exchange during that time. Who knows? We might even find out that, along with people who were originally from Norway or Denmark, Jorvik also had salmon from those areas too. Thank you all for listening to my talk about these amazing fish and their place in the archaeology of Jorvik. And I hope you've learned a thing or two about how important they were to the Viking people who lived here and to us today.